Hi everyone, this tutorial is about Bowtie. Bowtie is a read mapper that we'll be using to map next-gen sequencing reads against either a transcriptome or against a genome. So in this case, it's a starting point might be kind of like a blast search where we have a bunch of individual reads and we want to blast them to find out where they correspond to in the genome, what part of the genome they came from, or what part of the transcriptome they correspond to. The differences then between Bowtie and BLAST is that Bowtie is much, much faster, but BLAST is much, much more sensitive. BLAST can find sequences that are similar but quite different. Bowtie can really only find identical sequences or nearly identical sequences. Okay, so let's look at the data we'll be working with today. So we have first our reads, so these are next generation sequencing reads. So a series of reads. This is a FASTQ format. It's like a FASTA format. I'm not going to go through this in depth because I talked about it on the SRA tutorial. But basically what we're looking at here is a bunch of different reads, and each read has a read sequence. So this is a DNA sequence or an RNA sequence, and we want to compare that to some database to figure out where it matches to. Okay, so then the next thing we want to look at is a database. So this is the transcriptome, that is all the genes from the yeast Saccharomyces cerevisiae. So you can see it's a bunch of genes. Each one of them starts with an ATG, as we'd expect, and ends with a stop codon, again, as we'd expect. And so what we want to do is we're going to take the reads that we were just looking at, and we're going to map them to these genes. Okay, so you may remember in BLASTALL that there were two steps. First, we ran formatdb in order to prepare the database to be blasted against, and then we did our BLASTALL command. So similarly here, we're going to have two steps, and the first one is Bowtie build. So let me teach you how to run that. To run Bowtie build, you're just going to do Bowtie build, and then the name of our database. So in this case, I'm going to do it with copy because I previously ran this because it takes a little bit of time and I didn't think you wanted to watch it. So we're going to just type in the name of our file twice. So copy is just the exact same file we just saw. It's just a second copy of it. And so when I hit return, it's just going to start running. So what it's doing now is doing a bunch of math, packing in our sequence in a way that's going to make it optimally searchable so that it can be searched really quickly with Bowtie. And so you see a lot of stuff going by here. This is basically giving you status updates on how this is going. And in fact, we're almost done. It just would take probably another minute, but I'll let you do that yourselves. Once we've done this, then this is going to create some extra files. So here are the files it's created. All these ones that end in ebwt, and also these other ones that end in rev.1.ebwt. So you'll see it's created 6 files, each one that starts with the name of our initial transcriptome sequence, and then adds these different suffixes. So as with BLASTALL, when we formatted, it created some files, but we don't actually look at them because they're not readable. So if we look at this, it won't really make any sense to us. So that's actually, there's information in there, but we can't see it. So we're not going to look at these files, but we're going to use them when we do a Bowtie alignment. Okay, so then the next step is to learn how to use Bowtie. So Bowtie minimally takes 2 arguments, and it takes the database that we want to search against, and the name of our read file that we want to, the reads that we want to search against the database. And so if we hit return, it's going to spit a whole bunch of stuff to the screen very, very quickly, so that's not so good. So we'll go ahead and put it in a file so that we can take our time looking at it. So you can call it Bowtie out. And again, we're going to use this ampersand at the end so that we can get the command line back. Okay, so now the program is currently running, but we can start to look at what its outputs are. So I'm going to shrink the screen down a little bit so that we can get this looking reasonably. Sorry that my text on my computer is doing something funny. I apologize. It's not more readily readable. Okay, so what we're looking at here, so each one of these lines represents a case where a read has been mapped to one of the genes, where basically Bowtie has found that one of the reads goes with one of the genes that we're mapping it against. And so then our question is just figuring out what's happening for each of these lines. So as with a lot of bioinformatics files, it's just each line represents one piece of data. So let's go through it. So if we're going to have a read mapping against a gene, then the first thing we need to know is what the identifier or the name of the read is. So this is the identifier. That's like from the first line out of every 4 in our FASTQ file. And the second thing we want to know is where it mapped, what gene it matched to, and that's this information right here. So then the next thing we might want to know is at what position and on which strand did it map. So this 84 means that our read sequence corresponds to the sequence beginning with the 84th or 85th nucleotide of this gene, right? So it's mapped. So if this starts ATGAATGC, if we count out to the 84th position, then we would see that that's when it starts with the ATTTC, that's where our read mapped. This negative means that actually the read has the sequence that's the reverse complement of the transcript sequence in our database. So not the same sequence, but the reverse complement of it. So then we have the sequence of our read, and then the quality scores, which we talked about briefly in a previous tutorial. But they just, in this context, I don't use these quality scores. They just give us information about how likely we are to have an error at any position in our read. And then finally we have mismatches. And so that's mismatches. That tells us places where the read actually has nucleotides that are slightly, or that are different than the matching sequence in the transcriptome. And so here, actually, interestingly, we see lots of matches at position 0 and 1. I don't think we'll bother going into that today, but I'm happy to talk about that offline. And so there's our information. The identifier of the read, the strand it mapped to, the transcript it mapped to, the position in the transcript that it mapped to, the sequence of the read, the quality scores. I don't actually know what this is. And then any mismatches between the read sequence and the database sequence. So that's our tutorial on Bowtie. And you can see already that if you want to, then if our question is how many reads map to each gene, then we could write a little tally script similar to what we did in class that would tell us how many times we observed this in our 1, 2, 3, 4, 5th column, just with a split and a hash. But I'll leave that to you. And I'll leave off here with the tutorial. Hope that helps.